So it's the top of the hour, so we're going to go ahead and get started. Thank you all for joining this webinar today. It's going to be a very exciting time. What we're actually going to do is both in a sort of story form and in an interactive live way reproduce the results of a DNA sequencing discovery, really about finding a single causal variant for a novel X-linked disorder. Before I get too far into the presentation, just a little bit of housekeeping. I want to make sure that everyone is aware that on the right hand of your screen with the GoToMeeting dashboard, there will be a place called the little questions widget, and you can go ahead and start asking your questions at any time. I'll probably not pay attention to that as I go along until we get to the end, at which point I hope to give ourselves plenty of time to go through those questions. So whether it's questions about the story, the interactive analysis, follow-up questions with us, I'll try to get those addressed as much as I can. We have had, I think, over 300 registrations for this webinar, so it's a very successful, well, so far. I mean, I hope that if it comes out to be successful, but it's been great to listen to you all coming in. So we will also have a recorded version of this available after we kind of get through all this. It'll probably take maybe today or tomorrow. We'll be able to put that up, as well as we will have the slides available, so don't feel like you have to jot down notes or anything. So with that, we'll go ahead and get started. So what I'm actually going to do today is go through, both in story form and interactively, the reproduction of a X-linked novel disorder that was discovered by a research project done primarily by Dr. Gholson, Lyon and his team in 2011. And so on the story side, what I really need to do is step back and give you a little bit of history of how Gholson ended up with this project and how he went about doing it. And I think there's actually even more to the story than that, because this study really heralds in and as an example, or at least as a milestone, as we move towards using next generation sequencing in a clinical diagnosis form. Now, Gholson, to give you a little bit of background, came into practice as a psychiatrist primarily, and that's kind of where a lot of his training came from. He definitely has a molecular biology and genetics background as well, but in 2011 he was an attending psychiatrist at the University of Utah's Neuropsychiatric Institute, as well as having an academic post at the University of Utah. And so in that position, as an acting physician, a family approached him with sort of a tragic history in this family of having, over the past 30 years, 5 males who had a disorder and actually never lived past approximately 18 months. This was not necessarily, Gholson's bread and butter type of case, but as a physician, he felt strongly he needed to do everything he could to help this family. And he, with his genetics background, immediately recognized this as most likely a genetic disorder being inherited and passed through the family through the maternal line. It's only affecting males, and it seems to be an extremely highly penetrant, damaging disease and disorder, which when the males do have this disorder, they have some very pronounced symptoms physically, physiologically, and then you have infant death. So he went about this in a sort of standard clinical way, trying to get to a diagnosis. All genetic tests at the time did not lead to anything clear. The symptoms and the clinical presentation did not match any known disease. So he had actually, because of his academic post, the opportunity to turn this into a research project. And so, with that and with a little bit of funding and effort, he involved himself in every aspect of the research. He drove up to Ogden, the city in which the family lived, took blood samples, held the newborn child with the affected syndrome, took those samples back to his lab, got them sequenced, and was heavily involved in the analysis. And they did indeed find and functionally validated a single causal mutation that they believe and are pretty confident about was the causal variant in this disorder. So that led to a publication in the American Journal of Human Genetics. And so, all's well that ends well. But, actually the story goes on from there. And in fact, I find it most interesting in how that success leads onward as Gholson progresses, because essentially what happens next is the maternal aunt, essentially the sister of the mother of the child in this pedigree, in this family, comes to him saying, I know I was also, screened, etc., in your research project, but I just got pregnant. What are the chances that, am I a carrier? Am I likely to have this same genetic disorder passed on to my child? And this is where Gholson sort of ran into something he didn't really expect, which was because he did this research as an academic researcher, the sequencing was not done in a CLIA-certified lab in a manner that allows for the sequencing and even the Sanger sequencing that was done for the research project to be returned to patients. So as a clinical physician, he was not legally allowed to return that result of that carrier status to this mother who is pregnant and wants to know what's going on. And so obviously this puts him into an extremely ethically demanding and frustrating dilemma, and he uses that as sort of a case in point to make a much larger argument that, in his mind, this should not happen again. We should not be performing research on live human beings and not be able to return the genetic testing and the sequence data itself back to the patients. So to make that point, he actually was able to get an editorial into the publication Nature. So obviously a very high profile publication. He also has been on a couple high profile blogs such as Genomes Unzipped, and has been really making this this argument and this proposal to the research community across various different conferences, and I've had the opportunity to see him speak at a couple, and I've even met him a couple times before that. But this has really been a journey for him in making this point. So where does this all lead to? And as I have been preparing for this webinar, there is one thing also he makes alongside the point that data should be produced in a manner that can be returned to the patients. This is all part of a larger philosophy of Gholson's that we need to move away from paternalistic medicine, in which it's always we know best and we know everything and we give it to you on demand. In the case of genomics especially, we realize no single individual can possibly interpret all the complexities, and giving data back to the patients, who are really, in Gholson's mind, consumers or customers of this research, as they're participating in it, is the right call. So he is advocating for not just clinical grade sequencing to avoid these type of ethical dilemmas, but also more open access and collaboration across individuals. And that's how he contacted us, and we have been engaged with him to work on this project and reproduce these results. So as I was working on this, I also extrapolated this argument into a much larger context of, when are we going to get to the point where a family such as this one from Ogden, Utah, can walk into a clinic that doesn't necessarily have a research institute attached to it, or have a physician like Gholson who can turn a clinical case into a research project, and also might have a novel genetic disorder that they would like to have diagnosed and would really affect them as a patient, and from a clinical practice perspective allow for more options. When are they going to be able to take advantage of this great technology and have clinical diagnosis based on next generation sequencing be something that is expected, or at least has high adoptance? That is an interesting question, but it is important to keep in mind that, in some senses we are already there. There are indeed institutions, especially those that are close to research hospitals, that have a exome-based clinical test, and the physician can order it, and with that comes a genetic diagnosis. But this is still the exception and not the norm, and there are definitely challenges in both their cases and in any case of a clinic clinical setting trying to set up this diagnosis to get to the point where we can have robust, transparent diagnosis that are reproducible and meet all of our expectations of clinical grade and diagnoses. And it's not just about that initial CLIA certified variant list, and that's what a point that I go into in more detail in my blog. I put it up on Monday. You can go and search for Gabe Informatics or go to blog.goldenhelix.com to find it. I think we also put it in our chat queue if you're attending this live. But if you extrapolate out the remaining bits of the analysis toolchain beyond just that initial high quality variant list, we still have to work with a very fast changing field where algorithms change, sequencing instrument technology changes, the systematic biases of those instrument changes, and not only that, our research is continuing to progress in both providing more clinical or providing more associations between causal, highly penetrant variants and Mendelian diseases, as well as large common structural variations, and things like population catalogs continue to progress as the 1,000 Genomes projects move forward, etc. All those things mean that there is a lot of potential, essentially, reproducible issues that get involved in a research or even a clinical setting. And to address all those issues, we can turn to a staple of scientific advancement in a world of rapid advancement or rapid change, and that is to, for every finding, find at least one way or multiple ways to validate that finding, to reach that finding using orthogonal technologies or approaches or annotations or algorithms, etc. And so that's essentially the key of what we're trying to do today, is take the results of the analysis that was done in 2011 for this paper for Ogden Syndrome, and see if we can get to the same results using the information and best practices we have today. So we're going to use best practices because we want to really imitate the idea of an open, transparent set of processes that anybody could pick up and a clinical setting would try to use as well. But so to do that, let's go ahead and compare and contrast what we'll be doing compared to what was done in 2011 based on the annotations and tools of the time. It is worth noting before I go into much detail that although I talk about sort of the filtering process as if it was only one, Gholson also did a number of different essentially validations within his own study by approaching the problem from a couple of different angles to make sure that we were always concordant in the conclusion. Okay, so to start with, because this was obviously an X-linked genetic disorder, or that was the hypothesis, it was affecting only males. There were some males that were affected, there were some that were not, but it was and it was now in the third generation of the family. So this is why it was brought to Gholson's attention, is this is the first time it occurred at this generation, and so the family wanted to tackle what was going on. And in this case, what Gholson did is, because it was X-linked, they got a X chromosome exon capture kit. So rather than a whole genome exome capture, which weren't as quite as powerful at the time, but even at the point where we know that it's X chromosome, we can get more sequencing coverage here. This is just covering the coding regions or the exons of the genes that reside in the X chromosome. And the samples that were chosen was actually a very solid study design. The proband itself, unaffected brother and uncle, closely related individuals, and the mother and grandmother. And we'll see why that's important here, because what we're going to do is, or what they did, is really made sure that following the inheritance pattern expected by the hypothesis that this is a highly penetrant rare polymorphism or variant present in the proband, it would also mean that that variant would be carriers, or it would be in a heterozygous state in the mother and grandmother. So you can use that information as well as our controls to get to a very select set of variants that are candidates to be driver variants of a mutated or dysfunctional gene. Also, because it's expected that it is a potentially monogenic disorder, we would expect a single variant had to be non-synonymous to knock out the coding region of that gene, and we'll talk about more of that in more detail later. And finally, because this is a disorder that results in eventual death, we would not expect to see any adults from any of these larger consortiums or other public databases containing that variant, and hence we wouldn't expect to see the variant in these control databases. And finally, when they did find their candidate variant, they confirmed it with Sanger sequencing and expanded the number of samples that they sequenced at that site across the entire pedigree. And they also did functional validation to really confirm that that non-synonymous variant did indeed degrade the protein function of that encoding gene NAA10 with an in vitro assay. And they also, through the course of the study, found that that same variant existed in a second family, which was unrelated, and there was no affected males in that family, but they did find it in a carrier status in one of the females in that family. And finally, because this gene, really as they looked into it, did not have a lot of research, annotations, and publications around it, and yet it seems to be vital for overall functioning physiology of mammals. They are going ahead. Gholson has now moved to Cold Spring Harbor Laboratory and to a prestigious post there, and he's continuing some research with his group on looking into the disease pathophysiology of knocking out NAA10 in other model organisms. Okay, so that's the kind of the filtering process that was done then. So let's talk about what we could do to reproduce it using best practices today. One, we don't necessarily start from the very beginning because we can't go and get blood and resequence using, say, an Illumina HiSeq 2000. But we do, the sequence data is coming from extremely high read depth, high quality reads here, so it's going to be a very solid starting point. But for completeness, what we're actually starting with is 76 base paired single-end reads from an Illumina GA2. They put one sample on each lane, and the base calling was done with the standard Illumina software to get to the point where we have FASTQ files. Okay, so let's talk about how we would, pretty much in any study, looking for a rare, potentially highly penetrant variant, what would be kind of common best practices ways of going about that. First, you need to pick an alignment and variant calling strategy that reflects best practices. And so what we did here was used BWA and GATK. We ran that pipeline, which is actually very, there's a lot of intricacies to setting up those pipelines. So is actually run on a cloud compute platform called Seven Bridges Genomics, and the pipeline is set up to reflect the Broad Institutes, who publishes GATK as an open source tool currently. It's set up to sort of reflect their best practices. So it has all that good stuff like base quality recalibration, local realignment of insertions and deletions, etc. So that's the first thing, is to get to the point where we have a set of variant lists that are produced with the best practices pipeline. It's also good to note that because it's 2012, we want to do everything with the modern reference sequence. It's not that I would expect the reference sequence to change dramatically between HG18, which was used in the original study, and HG19. For a gene that is highly conserved, like we would expect it to be, those genes should be extremely stable from one version to the next. What matters more is now we can use all of the annotations that are based in the modern reference sequence coordinates. So the next step is to decide how we're going to do the post variant list. And again, this variant list, if it was produced in a CLIA lab like you can order from Illumina these days, is the starting point still of the interpretation process. Even if this variant list is CLIA certified, there's still a lot of choices to be made, and how do you get to that putative, validated, interpreted, annotated variant that might be the cause of a disorder. So what we're going to do is use SVS to annotate and filter using our latest public annotation sources. This is going to be an extremely transparent process. SNP and Variation Suite is a Golden Helix flagship product. It is a very powerful platform that's capable of very different types of genetic analyses, including copy number variation, RNA-Seq, as well as DNA-Seq, both in case control and small family analyses like this. We're going to use it as an annotation and filtering tool, do some visualization on it as well, but it's going to essentially use just these standard public annotations and filtering practices, and it'll be very transparent how we do each step. And finally, when we get to a list of interesting variants or get to a candidate variant we want to investigate, and really I recommend this for anybody who's doing research on next generation sequencing, don't stop at just looking at a spreadsheet or a list of variants. Always go back to the alignment data. There's two reasons for this. One, there are regions that are still problematic from a sequencing perspective. We have a short read sequence technology when we talk about next generation sequencing for large regions. Some platforms do allow for larger reads like Pacific Biosciences, but there's a trade-off of essentially read throughput and base quality and your length there. But when we're talking about an Illumina machine, we have 76 base pair reads. There are regions of the genomes where that just simply is not enough to uniquely align that read. And so there's going to be places where pseudogenes exist or other types of common duplications and trinucleotide repeats, etc., that are going to simply just be problematic from a variant calling perspective. The nice thing is, you open this up in Genome Browse, that is so apparently obvious as you look at it. You would slap yourself on the forehead and be like, all right, well, I'm not going to use that variant for anything that important, or I would need to do something dramatic like look at, in the case of our HLA region, you just simply use a different technology to get to those variant lists. And the second thing is, even if you do just find that you have a good quality variant there, all that genomic annotation information you might have been used in your analysis, you can use in a visual sense to really validate what you're looking at, get the big picture from the genomic sense. And all those public annotations that you would want, we try hard to make available, and you can stream those or download those directly inside Genome Browse. All right, so that's our reproduction strategy. And as I mentioned earlier, that's the story. Let's go ahead and show the practice. And by that I mean we can actually pop open SVS and do all these steps interactively through these various filterings and visualization steps. And our strategy here is to start from the very beginning, start with those VCF files that we ran through a BWA and GATK pipeline, and go about this as if it was our first look at the data. Now, it's again important to note, just like if when Gholson did his research, he went about it and found that he could get to the same result using 5 different approaches. We advocate the same thing. Although we're using this specific set of steps for filtering, we could reach the same set of conclusions using a different, population filtering technique than we're going to use by changing the order of things here. And in fact, we're going to actually do a filter based on the family structure. But just yesterday, to see what it was like, I started with only the affected child. Again, because this is such a rare disease, we expect the variant to exist, uniquely there. I started with just that child and got to a list of only 2 potential variants that matched the standard filtering techniques without looking at any of the brothers or the cases or the controls or the mother and grandmother. And out of those 2 variants, one of them was predicted to not actually, even though it was not synonymous, predicted to be tolerated at the level of changing the protein sequence. So essentially I got to the same single variant using a different technique. But we're going to go ahead and start with everything. And we're going to do, on first pass, an important filter that really you should do, or you should at least investigate, anytime you're looking at variants. Remember, when we call variants, we're not just getting to the point of genotypes. We have supporting information from the actual sequence data itself. That information will include things like the genotype quality and the read depth. And I'm going to show you what that looks like in histogram form. So I produced these histograms in SVS. If you wanted to get to these histograms yourself, they're actually extremely easy to reproduce. And in fact, what we did is we turned all the steps from this webinar into a tutorial form. That tutorial went live today. You can find it from our website. You can step through the steps and see how we performed everything click by click. And in fact, if you wanted to do it yourself, interactively run through it, Gholson has, excuse me, Gholson has set up a or approved a system in which you can request access to this data. It's still clinical data under a research setting, so it needs to be gated, but you can request access, and Gholson will be able to approve you if you would like to try to collaborate with him to validate these findings. So as I mentioned, we were able to get to these histograms. Let me just describe what these are in quick order. What we're essentially looking at is for all of the variants, and there was about 5,000 of them in our X chromosome, we're looking at the read depth, DP stands for depth, and the genotype quality across 3 different samples here. We have the affected male, the mother, and the brother control. So read depth is essentially saying at a given site how many reads support a support or essentially covering that site. And as you can see, the coverage here is very dispersed but extremely high for most variants. A lot of variants are covered by 40, 60, 80, up to 140 reads. These are going to be extremely high quality. There's very little chance for systematic or sort of error by not having enough evidence. Similarly, there are variants here that are just less than 5 reads covering those. Variant callers generally like to give you everything and let you choose what you want to keep. And that's what this step is about. We don't want to keep variants around in our analysis that only have a couple reads supporting them. They're going to just be too likely to be false positives and just simply not appropriate for this type of analysis. Similarly, genotype quality takes into account not just read depth, but other types of information from the alignment, such as the uniqueness of the reads being aligned to that region, as well as the base pair quality from the actual Illumina machine. In this case, you can see quite a few variants in all individuals are extremely high quality, and then there's a couple that are in the low quality region. So again, we're going to do a filter where we essentially drop genotypes that we find too low quality to continue. Now I'm going to choose, it may seem arbitrary, but again, we would expect exons that are in our axon capture to be covered quite heavily. These are starting to get into the frills of our coverage. Let's go ahead and drop things below 10. That should be a very conservative number, keeping most variants. And then on the genotype quality, we're going to drop genotypes below 20. It's also worth noting that there is a little bit more total DNA and a little bit higher genotype quality in the mother, who has 2 X chromosomes that are being sampled here than in the males, which only have 1 X chromosome. So you can see the genotype quality, the number that are high quality are a little bit higher, and you can see the overall histogram's a little bit larger. Okay, so what we're going to do now is jump on over to SVS, import these VCF files, and get started with this this process of filtering. So I'm going to go ahead and open SVS. I'm going to maximize it here so we're not distracted by anything. And SVS is a very, again, as I mentioned, a powerful suite of tools with a number of different modules to focus on different experiment types. But we're going to focus on just using that DNA-Seq module. So we're going to create a project here called Ogden Webinar Live. And there we go. And we're going to go ahead and say import VCF. VCF files are the product of the secondary analysis pipeline. That's what GATK is going to produce. And not only does it call variants for every file, it's going to put that supplementary information in there about read depth and genotype quality, as well as other things, as we'll see. So I'm going to go ahead and select all of my VCF files here. I gave the name of the files a base name. 2 options to keep in mind. Because we're having the VCFs produced in a manner where there's a single sample in each file is going to contain just the list of variants for that sample. So if one sample has a variant and another doesn't, there'll be a hole in the spreadsheet that needs to be filled in. We're going to fill those in with missings, which is just a personal preference, because we don't really know whether that hole is from low coverage or whether it was referenced there. For the purposes of analysis, it's all going to look the same, but just to keep in mind, missing is essentially meaning that sample is most likely just the reference matches the reference at that position. The other thing is, this is an X chromosome capture. Let's go ahead and only import data that's in the X chromosome. You would expect to see some variants sort of scattered across the autosomal regions. That's simply because there are regions that have high similarity between the X chromosome and other parts of the genome, and those other types of problematic or systematic biases of both the exon capture kit and the sequencing. So let's go ahead and then you see there's a couple different alternative data points with our genotypes, which is going to be our most important thing to start with. We're going to grab read depths and genotype qualities. Again, there's a lot of extra supplemental information here. Most of this is just a lot of statistics based on GATK's methodology, but we can focus on read depth and genotype quality. It's just going to take a second. Okay, so here we go. We have our genotypes that are imported for all 5 samples. Notice there's 5,000 columns. That means that there are 5,000 variants that are present in at least one sample. You can see some of these, in this case, only a single sample has an allele there. We can see what the reference is by expanding our annotations. And I'm going to point out also what are the details in those second 2 spreadsheets. So this is our genotypes. Notice again there's only one genotype per sample for these first couple columns. And let's look at the read depth. And sure enough, for those same samples, the read depth is only 2. So these are examples of those genotypes that we really want to drop before doing any more work. So let's go ahead and go back to our genotypes. And we want to drop these. To do that, we go to Quality Assurance, Genotype, set genotypes to no call based on additional spreadsheets. Say Add Spreadsheets. We're going to select our read depth quality spreadsheets. And essentially what we're doing is, based on the data of these spreadsheets, and we can choose a filtering criterion. We're going to say, the read depth here when it's less than 10. Let's go ahead and drop it. And the genotype quality when it's less than 20. Let's go ahead and drop it. And by drop it, I mean we're going to take those genotypes that are set and convert them to missing. So 2 things happen. Not only do we drop those genotypes, but we also inactivate columns that now don't have any genotypes in them. What that essentially shows us is we've gone from 5,000 variants imported in our all filter, and now we just have 3,000 left. So let's say OK, and I'm going to go ahead, and what I'm going to do is preserve this spreadsheet just as a top-level spreadsheet for our further analysis, and give it the name high quality variants. And I can close out everything else, and we'll start with this high quality variants when we get going. But you can see in SVS we essentially build up a history of all the things we've done. We've imported the genotypes, read depths, genotypes, qualities, and now we have our high quality variants. And let's go ahead and talk about our next filtering step. So what we're going to do now is take those 3,000 variants and apply the a filter to really look at variants that follow the inheritance pattern we would expect for this family. So remember, we have a 3-generational pedigree. There's a number of individuals at that 2nd generation, including some of the males that also had this disorder, and this is the first male in that 3rd generation that had the disorder. That male has a brother that was unaffected and a paternal uncle, which are absolutely excellent controls, because these 2 are in the pedigree. Both are children of what we would expect to be carriers of the variant for the mothers. This is the mother and the grandmother. Both of those are direct childs, but do not inherit the disease variant. So this is our configuration, and let's talk about what we would expect then from a genotype perspective. So because our mother and grandmother are carriers, we would expect them to have, of their 2 X chromosomes, one of them to have the alternate allele, alternate to the reference, essentially what we mean there. You could also just call this a heterozygous genotype or a polymorphism in a heterozygous state, but essentially they have one variant that's completely matching the reference, which would result in one copy of the causal gene that is functional and one that is non-functional. And because of that, we would expect that this gene is haplosufficient. In other words, even with one of the copies knocked out, we have a functional gene that allows for the individuals to reach full health. The second thing we can count on then is that the 2 living males inherited the working copy of those genes and hence have a reference at the variant site that we're looking for. And finally, we would expect the affected male to have the alternate allele. Now notice there's only one allele present here, not 2. The reason being because they're males, they have 1 chromosome. We would expect them to be in what we call a hemizygous genotype state, where they only have a single allele. In this case, it would be alternate. Now, even though it should be hemizygous in biology, the genotyping or the variant calling algorithms don't necessarily pay attention to sex. They're going to call it as they see it. If they see if they see a place where there's a heterogeneity of alternates, they're going to call a heterozygous genotype. So we would expect the variant to be a homozygous genotype from a filtering perspective. So let's go ahead and do that step now in SVS. So there we have our spreadsheet that has our high quality variants, which is our starting point. And we're going to go to Select, Filter by Sample Genotype Patterns. That's just a clever way of saying we're going to look at any type of variant site that matches that set of restrictions on what we would expect the genotypes to be, for our individuals. Again, our affected male, we expect it to be alternate, homozygous alternate, because we're not distinguishing between hemizygous and homozygous. Mothers and grandmother, we expect to be carriers, to have that in a heterozygous state. And the brother and uncle, because there are controls, they should be reference. But remember, we filled our holes in with missings, so missing also represents reference here. So as we apply that filter, we take it down from 3,000 variants to a meager 67 that match that inheritance pattern. With that level of, this is why an experiment design can be so effective at getting to a robust conclusion, because even with just that information, we have a very manageable set of variants remaining. So let's rename this as our high quality candidate variants. One other thing I want to show here is let's look at how these variants, you can see their individual positions and reference alleles. Let's see how they actually look in genomic space. We're going to pull up what we call a variant map, which is essentially just a representation of any place that their genotype is non-reference, and plotting that in genomic space. Here's our X chromosome along the bottom. You can see there's really only two regions where this inheritance pattern, where there's variants that match this inheritance pattern, and this is completely logical, as we would expect only a couple recombinations of the mother's X chromosomes per generation. There's essentially only a section here on the P arm and a section here on the Q arm that match that recombination being inherited by the male, and the recombination for 2 generations was not inherited by the brother and the uncle. So this actually shows us, what regions are essentially inherited and risked to the male, and this was something that in the paper Gholson also orthogonally found that there was 2 regions through haplotype analysis that were inherited in a way that could contain the disease variant. So there we have our high quality candidate variants. Let's go back to our next filtering step to see what we do next. So we move from now our 67 variants to applying a look only at variants that are non-synonymous in coding regions. So I explained this a little bit, but to go into more detail, this is what a coding variant classification report looks like, where essentially for every variant that falls within the gene annotated regions that are in coding for amino acids that are part of the total protein sequence, a variant that is inside of that region will come up here, and it could be potentially deletions, insertions, single nucleotide variants, and they could potentially change the protein sequence or not. Because of our redundancy in our amino acid encoding from nucleotides, there are certain variations that even though they go from like a T to a C, for example, in this position, they do not change the protein sequence, and we want to filter those out. We want to look at only coding variants that are non-synonymous. Again, we don't care if they're necessarily insertions, deletions, or single nucleotide variants or nonsense mutations, but we want to filter out the synonymous variants. Let's go ahead and jump back to SVS. Start with our high quality variants. Go to Analysis, Variant Classification, and I don't care about the gene counts. What I do want is a report of all variants and their classification, and a list of coding variants. So it's going to produce 2 spreadsheets. And I'm actually going to close that first one, which is the coding variants, and talk for a second about that list of high quality variants. But notice we found only 12 variants that were coding. So now this is the list of all high quality variants and how they fit into gene annotations in general. If we click on this classification column and say value count, we can get a feel for the fact that there are only 12 here, but there's, what's going on is that exome capture expands across just exons to show, some variance in the UTRs and introns and up and down. They're focused or centered around exons, but they definitely are going to catch variance outside of just the exonic region. So if we look now at our coding variant report, we can see there are 12 variants. There are some that are synonymous. We don't have any indels that have reached this phase. Given the sort of ratio of indels to synonymous or single nucleotide variants, that's understandable. They're definitely less common. We want to, and I went to right click and I said activate by category. What I'm going to do is actually use this spreadsheet as a filter. I'm going to grab these 8 variants that are now active, 1, 2, 3, 4, 5, 6, 7, 8, and I'm going to go ahead and say select, apply that set of 8 variants as a filter to my genotype spreadsheet. Now that was my parent spreadsheet. I always want to work with genotypes when doing further analysis, so I'm going to use this as a filtering step to take that set of 67 variants down to 8 synonymous, and go ahead and preserve that spreadsheet as a node and give it a new name of that are non-synonymous. So there we have our non-synonymous variants. Let's go ahead and look at our next step, which is to pull in functional and population information. And I'm not going to spend too much time on this, but there are definitely choices here on what type of population information we want to use. And when I say population information, what we want to do is say, hey, this is a variant, but does it exist fairly commonly in the population of humans at whole, or within the individual ethnicity I would expect to compare against? So first of all, the 1000 Genome Project is probably the best known variant population catalog. The nice thing about that is their variants are based on whole genome sequence, so you have variant coverage across the entire genome. They only have about 1,092 individuals. I should say only. That seems like a large number if you think about whole genome sequencing. And they also update their catalog quite frequently. So when you talk about filtering, you always need to be specifying which specific version of a variant catalog you're using. There's also dbSNP, which is a very probably the longest running catalog of known variants, and it also has population information in it. But it is worth noting that most of that population frequency actually comes from older versions of 1000 genomes. So if you're going to actually filter by dbSNP at a frequency level, you're essentially filtering by a 2011 release of the 1000 genome frequencies. And finally, there's the this excellent catalog, which is going to be appropriate for us today. It's published graciously by a consortium that's not publicly funded, but it is the National Heart, Lung, and Blood Institute. I should probably clarify, I'm sure some of their funding is public, but they were no means obligated to release this data, but they did it for sort of the community benefit. And they have over 6,500 individuals across European and African ancestry, and their variant catalog is based on exomes only. Excellent for this type of analysis where we have exomes, highest catalog of number of individuals. Also worth noting that they have complex diseases within their cohort. So it's not that these are perfectly healthy individuals, but they are individuals who have reached the age of consent, etc. The other thing that's going to be very useful is the genotype counts, not just the allele frequencies, are available in this catalog. And I'll show you why in a second. Finally, alongside just population information, one thing we have today that we didn't necessarily have with as much detail in 2011 is a lot of tools that focus on predicting whether those non-synonymous variants, remember we've gotten to 8 non-synonymous variants so far, are likely to actually be damaging to the protein's function. So even though you might change an amino acid in a protein, that doesn't necessarily mean that that protein can't go about its day doing everything it would like to do. You might have a very large protein, maybe it's got some subdomains that can tolerate a little bit of mutation, and so you can actually look at things like conservation scores and other types of clinical information and build giant predictive models that essentially predict whether a non-synonymous variant will degrade the function of a protein and damage essentially the function of the protein. So the common ones here are SIFT and polyphen 2. And there's this great catalog that's been published called the Database of Non-Synonymous Functional Prediction that we're going to use that actually pulls a lot of these things together in one great place. And you also have things like population or conservation scores letting you know if a single base is highly conserved across mammalial species. And one thing to note, though, is that these are very complex, systems biology level pieces of information we're trying to integrate to make these predictions, and they're just simply not going to be 100% accurate. In fact, I think there's been a number of studies showing that if you sample these and do a deep level functional validation, only about 80% of the predictions are accurate. Again, that's a pretty good number for something so complex. It's great for annotating variants. It's great for prioritizing which ones you're going to go and do some functional validation on, but you might not want to use this as a primary filter if it potentially would throw out some false positives at you. So before we jump into anything else, let's go ahead and take our non-synonymous variants. Here's our 8, and do those 2 annotation steps. So I'm going to go to Select, Filter by Annotation, Variant Frequency Catalog, select that 6,500 exomes. There's previous release in which the cohort was smaller, but we're going to use the latest release, 6,500 exomes. And I can actually use this as a filtering tool if I wanted to. It would drop variants that were below a frequency. We only have 8, so let's just use this as an annotation tool, and we'll pull up a spreadsheet of all of the matched variants. And when I say matched, what happened was only 7 variants of our 8 were actually discovered in there, meaning that there's one variant that is completely novel. So we'll come back and look at that in more detail in a second. The second thing we're going to do is say Select, Filter by Annotation, Filter on Non-Synonymous Functional Prediction. And again, we could use this as a filtering tool. We're just going to do this as an annotation tool. And here we should expect, because we're looking at non-synonymous variants, that we have predictions for all of them. In fact, we do. We have 8 non-synonymous functional predictions, these SIFT and polyphen predictions, and we'll use that together. So what I like to do from here is actually jump back to my coding variant classification report, create a spreadsheet here of just these 8 non-synonymous variants. Because this has that information about the gene, the transcript, and the protein changes in it, let's use this as an aggregation of our annotations for our remaining candidate variants. I'm going to go to File, Join, and essentially join in these 2 annotations. I can join in the DB or the NHLBI 6500. Remember, this one didn't actually have all 8, so I have to say if there's unmatched rows, go ahead and keep those. So I keep my full set of 8 variants. So now I still have 8. And while we're at it, let's join in the non-synonymous functional prediction. Go ahead and say keep. Okay, so now we have, just close out the rest of these spreadsheets, my set of variants and all those annotations all in one spreadsheet. We scroll on over, we can see these allele frequencies, and we're going to use the European allele frequencies. Let's go ahead and sort this in an ascending order. So we have the rarest ones on top. Sure enough, there's that one that's completely novel. It doesn't have an entry in the database of known variants. And you can see actually quite a few of these are common to the point of being 40, 14%, 3%, 2%. So this one's interesting. It's below 1%, and that's actually very rare, especially in such a large cohort of just Europeans. So we might want to keep an eye on that. But as we scroll over and look at those genotype counts, as I mentioned, so this is that rare, and this is the genotype counts. So the reference here is a C, and notice again the hemizygous genotypes are going to be the ones from males where there's only one allele. Just as I was mentioning earlier, females are going to have 2 alleles in the X chromosome. So there are a lot of people, 1,870 males in this cohort that have a C, but there are 2 that have the variant here. So if you have a variant present in 2 males that made it to the age of being in the study, unlikely, in fact, pretty much rules out this as a potential variant for this disease. You're not going to have that variant that's going to cause this infant death also be present in healthy adults in the hemizygous state. So let's go ahead and scroll over some more to that functional prediction stuff. And again, we have a number of functional prediction tools all kind of brought together: SIFT, polyphen, mutation taster, and some conservation scores. Remember, our first row here, our top of a list, is the rare or the completely novel variant. It's predicted as damaging, probably damaging, and disease causing. So essentially a full suite of predictions saying that this is likely to be a variant that knocks out the gene function. It's also highly conserved by the two different conservation scores. Just to give a feel for what's going on, the second variant is predicted as damaging, but doesn't have predictions on the other two. So a number of reasons why. Again, there's a lot of annotations that go into these predictions, but you can also see the other variants that were higher frequency are predicted as benign, tolerated, although you can see again that these things never necessarily line up on all predictions. This one has a different prediction based on SIFT and polyphen. So let's go ahead and see, scrolling back over, what this variant is, this novel one at the top of our list. Well, indeed, it's the variant in NAA10, so we have a single unquestionable result here. It is the variant that was shown and through functional prediction or functional validation to be the causal variant of this disease. So I'm going to copy this label. It's essentially the chromosome and position of it, and we're actually going to follow this up in Genome Browse. So what I'm going to do is I'm opening Genome Browse here, and Genome Browse is Golden Helix's free visualization tool. It's absolutely free. We're going to do a lot of great things with this tool in the future. Feel free to jump on board and start playing with this. We have a lot of annotations you can bring in, but it's designed and really optimized around doing an interactive set of analyses and validation. Because I'm running all the way up to the end of the hour, instead of going in and jumping into creating this project from scratch, I'm going to open up the project that has all of the files. I'm going to not save my project here. All of the annotations I would like to explore here. This has the OMIM genes track, a list of segmental duplications. Those are common areas to keep an eye out, like I said, when you're looking at variants that potentially don't validate. Here's our 6,500 exomes, 1,000 genomes, our reference sequence, our affected male, the mother, etc. You can see at a whole genome level, the data is definitely focused at the coverage, peaks up above the baseline at the X chromosome. Let's go ahead and double click on X, and we can see the coverage here. The nice thing about Genome Browse is that it's so intuitive. You just start rolling your mouse forward, using a mouse scroll zoom, and you can get down to the place of seeing individual genes, highlighting those, seeing individual variants show up in these variant catalogs, and seeing as you double click on a gene, the individual reads showing up. In this case, this is an all reference gene. You can see all this annotations, it's an OMIM, etc. Let's go ahead and jump to that location for our variant we're validating, and sure enough, here it is. You can see where it is in the amino acid sequence, and you can see where it is in the gene. There's no, for this exon, I'm going to kind of center this exon of NAA10, there are no other variants in either of these catalogs. I'm going to go ahead and just hide these so we have, and there's also no segmental duplications. So we have fewer things we're looking at. It's also in OMIM gene, meaning this gene has been associated with other Mendelian diseases in a diseased state. And so the information here is very, very clear. We have 56 reads supporting the G. There's nothing else that would imply that this is a read we wouldn't trust. I can zoom into the base level and see the individual reads, and it's very clean. There's reads above and below the strand. I could zoom out and see the alternative exons. In fact, let's go ahead and zoom out to the entire gene level, and you can see there's no other variants for this sample. I'm going to go ahead and hide its pileup and show just that we have the mother and the grandmother, which are essentially in a carrier state. Those variants are showing up clearly as heterozygous, as we would expect. So the variant calling looks very clean here. Let's add in also, I'm going to unhide the unaffected brother. And there's, outside of the exon, another variant here that was inherited from a heterozygous state to here. So you can see that there is essentially two haplotypes, as we would expect. From our chromosomal rearrangements or from our recombination, the brother got a different set of haplotypes, a set of essentially inherited the chromosome that's represented by this heterozygote here, and the affected male inherited the chromosome represented by this heterozygote. So that's essentially the type of thing we want to do with genome browse. We can also look to the fact that there are very few, if any, variants that fall inside of the coding regions of this gene, implying that this gene is pretty highly conserved. In fact, this variant that does fall in the coding region occurs only in an African individual, and it does not actually occur in there's no T, right? There's no hemizygous, so there's no male that actually has this variant, also implying that this gene is extremely important. And indeed, this is the functionally validated result. So there we go. We've used Genome Browse to kind of get to the result we were hoping, and we can be very confident that this is a high quality variant. And in fact, Sanger showed it to be true, but to the point where you have this many reads supporting it, Sanger sequencing, I think, is eventually going to be antiquated, or at least not necessary unless you are less confident in this case. So let's go ahead and kind of wrap up our presentation here. We've gotten to our one variant in NAA10, the one we would expect that matches the variant published, and we did it through an orthogonal set of highly transparent set of steps using SNP and Variation Suite and Genome Browse. And so, I mean, that was the goal of this. And as I mentioned earlier, we have a we have a tutorial that steps you through all the steps in which we took to get to this point. So that gives you your own ability to validate our steps, but also potentially learn from that. Although we did a lot of things here that are specific to the study, pretty much any type of highly penetrant monogenic disorder hypothesis is going to have some very similar aspects. You're going to look at the family pedigree, whether it's trio, quads, or whatever you have. You're going to look at tightly matched controls, do filtering. We have a number of other tools specifically for looking at rare recessives. And if you were doing things not just for novel diseases, but you still think it might be rare, you can do filtering where we just did annotation and get down to rare variants. So with that, I'm going to open it up to questions. We still have number of people on the line, but what I'll do is take my best shot at some of these as we go. Feel free to also follow up with me or the support team at support@goldenhelix.com if you have further questions and you would like to have a discussion about your thoughts on those. Okay, so I'm going to try to pop out my question dialog so I can see questions here. All right, so one of the questions is, what is the difference between RNA-seq and DNA-seq? Very good question. RNA-seq is actually sequencing mature RNA that is present in the cell at the time that the sample preparation is done. You're creating a library for sequencing. So in this case, this is an X chromosome capture kit. What you're going to do is you're going to take blood, you're going to extract out, you'll purify the DNA, use a way of shattering that DNA into small fragments, and then the capture kit's going to pull down based on little tiny matching primer sequences, pull down any reads or any fragments of DNA that are covering the intended regions, in this case the X chromosome. RNA-seq, it's very difficult to sort of do targeted RNA-seq. You just go for the whole transcriptome. What you end up doing though is trying to reduce the amount of ribosomal RNA from the sample, pull down the mature RNAs either through a through a couple of different library prep techniques. But in that case, what you're really looking for is not so much variation from the reference sequence, but a very large distinct variation in the expression level of genes. That is a complementary tool for this type of analysis, because you might want to say, for example, was this gene expressed at a lower level in the sample or in the blood of the affected individual versus the unaffected individual. And you could also do that on a targeted basis using something like a Western blot plot, which I think is what Gholson did here. Okay. Next question, if the disorder is not rare or completely novel, can I filter out the variants that are present in dbSNP 1000 genomes? So it's a very good question. I think I did allude to that just a little bit. But the answer is absolutely. You can, we used those databases as just to annotate our few remaining variants. A very common workflow is to start with your highly qualified or high quality variants. From there, use something like 1000 genomes, dbSNP, or both to filter down to a list of rare variants where you're just using that allele frequency information. So you want to look for variants that are less than 1% or novel in the population. Next question, can we upload a BAM file here? I think that refers to Genome Browse, and as I said, I didn't have time to build up this project from scratch, but from our add dialog, it's very easy to add in your own BAM files. So by adding them, if you haven't before, you will just throw them in there as long as you have the kind of required supplementary files. If you don't have them, we will compute it on the fly, etc. It's a very easy to use tool, and it should do everything for you in terms of getting your BAM files ready to be visualized. Yep, so next question, prediction tools usually provide conflicting prediction results. How do you resolve one prediction as benign while another predicts as deleterious, right? So in fact, we were looking at that exact case in our report. There was one of these that is benign versus deleterious. And this is certainly, at the end of the day, if you're doing clinical genomics, you don't stop here. This is just informing you on which candidate variants you want to continue on to a functional validation, etc. So, I read a recent paper and I talked about this in my blog, by a group at Duke, by Goldstein's group, and they did 12 exomes for 12 cases that had not yet reached a diagnosis. 6 of those they reached a diagnosis, and they showed their own way of making sure that they got to a diagnosis. And it wasn't just by filtering. You use something like this to give you a level of confidence of which ones to follow up with. Then you can do a Western blot plot to see that a protein in the case of one of the males in that study was completely annihilated. You can see it compared to the mother and the father. It was a trio study. There was simply no levels of expression of that protein in their blood. And so similarly, when there are conflicting results, again, complex system biology is going to produce that when these have different sort of methodologies to reach these results. People have proposed using a voting strategy where you have 5 tools and the ones that majority wins. That's all good for getting down to a list of variants, but ultimately you're going to have to take it back to the lab to get to a conclusion that holds up at a diagnostic level. Yep, there's a great question here that GATK calls multiple samples at a time potentially, and let's see, is there any reason you have this set up as a single sample? The reason is simply essentially bioinformatic convenience, I guess. The variant caller that calls multiple samples together is a more computationally expensive, runs on a bigger machine, takes longer, takes more memory, etc. I actually do prefer it as well. If you can at all possible, put a trio into the consensus genotype caller where for every single variant it wouldn't fill in it with holes. You would have a single VCF file that fills in with reference where there is supporting evidence for a genotype, and with missing when there's no reads to cover that genotype. That is obviously preferable, and but at the end of the day, genotype callers are still going to have problematic areas, so you can't just also treat that as a silver bullet either. There still has to be follow-up in genome browse, etc., to really validate that any putative variant. Another question is that sequencing module that I mentioned SVS has, does it handle whole genome sequencing, exome sequencing, DNA methylation, RNA-seq? Yeah, so 4 questions there. Yes, it handles whole genome sequencing. Yes, it handles exome sequencing. We have customers doing DNA methylation in it. I wouldn't say we have all of the workflows there fleshed out to support that as a first-class experiment type, but we definitely do support RNA-seq differential expression profiling experiments. So in whole genome sequencing, a lot of these steps look exactly the same. Again, you're just going to have a lot more variants. We can open spreadsheets with 30, 40 million variants as columns. It still opens in a very memory efficient manner. As I mentioned, SVS is a very robust and powerful tool, and this is sort of our bread and butter stuff. So whole genome sequencing on small samples or large number of samples is very possible. Do you have a track that shows genes predicted to be haplosufficient? That's a really good question. I've been actually looking for the best, or I guess a good best practices track to display that information. I do think that that information would be extremely useful in this type of study as well as others. So I'm actually planning on adding that as a track in our next round of data curation. So the answer is not yet, but coming. So next question, this is probably a looking forward one, which is a great one. If we know that genotype quality and read depth for sequence data will improve in the future, do you think that individuals who have their genomes or exomes sequenced today will have to submit another sample for resequencing? I think this is really asking the more hypothetical question of, could you ever just sequence once and use that data forever? My feelings is, maybe, but at the end of the day, it's really about what you're trying to accomplish in your analysis, or really what the experiment calls for from a I guess assay solution. If you're talking about rare, highly penetrant variants in coding regions of genes, heck, exomes these days are cheap. They're extremely high coverage. I have my own exomes from 23andMe that I've presented on before and will do again. I have every confidence that if I were to look at a variant that looked as good as this one in Genome Browse, there's no reason I would ever doubt that that exists biologically in my DNA. At the same time, if you're looking, potentially asking the question about, does a sample, do I, or if this is my exome, have a copy number variation, this is not the tool to answer that question. Is whole genome sequencing the silver bullet? Similarly, there's going to be specific types of genomic alterations that it's not going to be the silver bullet for. Copy number of variation with whole genome sequencing is definitely improving, but maybe not even as good as the cheap cytogenetic arrays. Similarly, there's going to be regions in the HLA that sequencing will never get that correct without a lot of very specific algorithms or approaches, or having a much better read length or other types of properties that would help with those types of problems. So there's no single tool that will ever give you something you can use in all cases, but certainly sequencing is getting to the point where you can trust high quality variants for the specific types of genomic alterations they're designed to test. Can you analyze paired samples, i.e. tumor normal? That's a very good question, and actually leads to an ongoing, in our goal to present these types of webinars in an ongoing fashion. We want to continue to expand the types of analyses we do in an interactive session like this. So we do plan to do a tumor normal pair analysis of an observational cancer study and hope to do that in the next couple months and demonstrate similarly how SVS and Genome Browse works with that. Not the nice thing, but the interesting thing or the important thing to really spend time with there is the fact with cancer studies, you do not have these clean heterozygous variants that are called with perfect read distribution of half of reference and half alternate. That's simply because you can't get a sample of a single cell that easily sequenced, so you can't get a sample that contains pure cancer cells and no normal cells. So that heterogeneity of the cell assay results in the fact that a valid cancer somatic variant might occur at 5% of the total read depth. So all that type of stuff is very important and has a different approach to be used in SVS and Genome Browse, and I'll be talking about that in further webcasts. What is the evidence of selecting the cutoff for high quality variants of 10x and Q20? As I mentioned, there's no way to say this with complete certainty that you will never remove a potential, candidate variant. So essentially have false negatives by doing this type of filter. What I can say with certainty is if you don't do that type of filter, you will have an enormous number of false positives from those genotypes based on one or two reads. Now, in the case of R, again, this is the distribution of reads. If you had genotypes over here, would you trust them? Why would you trust them when you know your genotypes or your variants are going to be centered around these exons, which by the nature of the assay are going to have high coverage? So those filters are specific to the experiment and specific to the sequencing choice here, and that's why you look at the histograms. You look at the actual data instead of making those up from complete scratch. I think that those read depths of Q20 and 10x are actually very conservative. Reads of 10x are to the point where it would be very questionable to use those for analysis, but you can certainly keep allowing for more and more reads to come into your analysis and then weed through the false positives at a later basis. My only emphasis is that be aware that without doing some sort of filtering, you're going to have extremely low quality variants following through your analysis and potentially confounding or just getting in the way of finding things like this high quality variant. All right, this is I think that it's going to wrap it up for us today. There's just a couple more questions, but feel free to follow up with me online on those or offline with those. I appreciate everyone joining the webinar. We will again have a recorded version of this webcast posted online. Keep an eye out for that in your inbox or come seek it out at goldenhelix.com. And I hope this has been an informative discussion for you. And so, continue to interact with us, if you will, through our blog and through our support lines. We look forward to collaborating with further individuals and researchers in the future, just like in this great case of showing how we were able to reproduce a finding done by Dr. Gholson Lyon at University of Utah on Ogden Syndrome. So thank you very much. And with that, we're going to close the webinar.